Very good morning, my friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So we are pleased to inform you that our Government Degree College for Women, Jagityal, and also the RBVRR Women's College, Hyderabad Autonomous, and SRR Government Arts and Science College, Karimnagar. These tri-colleges, along with TGCGTA, Telangana, is going to conduct wonderful program that is national level online coaching for plant sciences and CPGET-Botany. So in this regard, today's eminent resource person, Dr. Rani Madam Garu, Dr. Rani Komidi Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous Siddipet. Madam is going to explain one of the important area in plant physiology, photo-oxidation of water and photophosphorylation. Madam is going to explain. So, Madam, please welcome and start your session, Madam.
Okay, sir. Thank you, sir. Are you able to see my screen, sir?
Yes, yes, Madam. It is clearly visible. Please, you start.
Okay, sir. Thank you. Okay, yesterday we'll discuss about that in the photosynthesis. So the photophosphorylation. In photophosphorylation, it is completely the content of that photooxidation of water, photophosphorylation, and the mechanism of electron transfer, types of photophosphorylation. So yesterday, the two topics, that is cyclic photophosphorylation and non-cyclic photophosphorylation. We will discuss about completely cyclic. In non-cyclic, just to let me clear some topics. So this is completely the photophosphorylation, Nana. So in the process of photosynthesis, the phosphorylation of ADP to form ATP using the energy of sunlight, that is called photophosphorylation. It is divided into two steps. So in that first one is the cyclic photophosphorylation. Yesterday we'll discuss completely. And the second one is the non-cyclic photophosphorylation. So in the non-cyclic, while coming to the non-cyclic photophosphorylation, it involved the two systems. So almost we discuss some involving of the photosystem one also. So let me complete the photosystem or the non-cyclic photophosphorylation. So in the non-cyclic photophosphorylation, completely it is Hill and Bendall proposed the scheme of photosynthetic electron transport in 1961. So it involves the both photosystems 1 and 2. So these both will acting in series. They connected by electron transport mediators. So it's like look like a drawing this scheme on the redox scale. So the look like a Z letter. So the letter Z, and hence the scheme is often known as the Z scheme. So in this completely, I will show another diagrammatic. Just wait. Photosystem 1 and 2 look like a Z scheme. So this process begins with the absorption of light energy by light harvesting complex. So this is the complete photosystem 1 and 2, both will involved in this process. So it is transferred to P700. So this is the reaction center by resonance transfer. The light excited P700, it expels the electrons. So this is completely P700. It will, when it is undergo the excited state, it will just release 2 electrons. The electrons are, first primary electron acceptor is the iron sulfur protein. So in this, the first electron passes through a chain of intermediate electron carriers to a soluble pyridoxine that is presenting in the stroma. So completely we'll discuss that the electron finally proceeds through the 3 membrane bound pyridoxine. So that is completely, they contain the iron sulfur protein and some soluble pyridoxine also. In this way, for reduction of the 1 molecule of NADP, 2 electrons are required, and these 2 electrons are provided by the pyridoxine at a time. So the reduction is catalyzed by almost, as I told you, that is pyridoxine NADP reductase. So in this, the enzyme pyridoxine NADP reductase, apparently it involved in matching the 1 electron chemistry of pyridoxine to the 2 electron chemistry of NADP. So completely, this is since the expelled electrons, they ends up in the reducing the NADP plus, so it results in the electron deficiency in P700 reaction center. So commonly it is referred as a hole. So this deficiency is filled up by electrons. They are derived from the photosystem 2. So up to here, we'll discuss yesterday. In this, the first system, that means the photosystem 1, it will gives the electrons are driven from water pass to a mediate by pigments in the photosystem 1 and 2. So in this, the 2 light reactions, they are interacted in the photosynthesis. So it research on the dependence of photosynthetic rate on the wavelength. In this, the first photosystem 1, it will produce the ATP and 2 NADPs are the finally assimilation power of the photosystem 1. Okay. So while coming to the photosystem 2, the photosystem 2 are the completely, it is gives the photolysis of water. In the present, in green plants, the photosynthesis, it is mediated by 2 kinds of light reactions, Nana. So in that completely, the photosystem 2 is the chlorophylls are, just wait, photosystem 2. So the photosystem, it is 1 minute. So the photosystem 2, that is the reaction center P680 nanometers of the wavelength. That is the photosystem 2 reaction center or pigment system 2 reaction center. So the energy trapped by light harvesting complex, so it is transferred to P680 by the resonance transfer. So in this, the excited single electron in P680 is transferred to the PHYSIOLog. So the reaction center, the pigment system 2 reaction center, our primary electron acceptor is the PHYSIOLog. So the electron deficiency in the P680, it is obtains electron from photooxidation of the water. So this is very important. The photolysis of water or photooxidation of water is occurring in the non-cyclic photophosphorylation. So in this way, the electrons are removed from water by manganese containing the oxygen evolving complex. So this means completely water molecule is catalyzed and the water is undergo the photolysis, gives rise to oxygen plus 4 protons, 4H plus oxygen is in the end product. So in this way, the electron deficiency, it is completely from photooxidation of water. The electrons are removed from water by manganese containing the oxygen evolving complex. So it is located along the inner side of the thylakoid membrane. So the mechanism of water oxidation, that is not clear, and it is in the formation of the oxygen. That means 2 water molecule, 2H2O gives rise to oxygen plus 4 protons. So in this, the oxygen evolving complex cycles through a series of oxidation states while removing the protons and electrons from the water molecules and finally release the oxygen into the inner thylakoid membrane. So this is a very common equation. It is occurring in the only photosystem 2. The photolysis of water is, just I will go the first slide, the photooxidation of water. So the PS2 reaction center P680 nana, by transferring the electron to primary acceptor, it becomes the oxidized state. So the overall process is called the photooxidation of water. It requires the presence of Mn+2, Mn++ calcium plus and Cl minus chlorine. So a water oxidizing enzyme and an unknown substance, that is the Z. So it is believed that oxygen evolves as the oxygen gas. So electrons are accepted by PS2 reaction center through unknown substance. So unknown substance means almost yesterday we'll discuss that DCMU also one of the cause for that. The DCMU, that is chlorophenyl dimethyl urea. So it is an eligible and it is an herbicide, Nana. DCMU also, it inhibits the complete photosynthesis. So it was first introduced by Bayer in 1954. That means the DCMU also, it inhibits the photosynthetic process. It blocks the plastoquinone binding. So completely the plastoquinone binding site of the photosynthesis too. So it completely disallowing the electron flow from where it is generated in the PS2 photosystem or pigment system to the plastoquinone. So this interrupts completely DCMU also, it interrupts the photosynthetic electron transport system in the photosynthesis. So it is main cause. That means the electrons and protons are the complete power of the ATPs, NADPHs also formation of that to inhibit this DCMU. It is one main reason, and almost the electrons are accepted by PS2 reaction center through the unknown substance, and protons temporarily stay in the thylakoid space. So the high energy electrons that leaves PS2, they are captured by the quinone or Q, which sends them to an electron transport system, it consisting of the plastoquinone. And completely these all are called as a electron carriers or electron transporters: plastoquinone, cytochrome B6 and cytochrome F and plastocyanin. So these all are called as a cytochrome complex and plastocyanin. So every time, just you can observe here, Nana, in the diagrammatic way, this is the electron transport chain photosystem to water molecule is undergo the photolysis. So it gives the oxygen and protons. The deficiency protons are accepted by through this photolysis of the water. So 4 electrons are come from the photooxidation of water. So the chlorophyll, it excited to the primary acceptor, that is the Q, and electrons are passed through the electron transport chain. So in this cytochrome B and cytochrome F. So in this, just you can see here, this is the complete Q, plastoquinone to electrons, from plastoquinone to cytochrome B6 and cytochrome F complex. So in between, these all are called as a pool of that electrons. So after that, electrons come from the plastocyanin after it is reached to the photosystem 1, that is the P700 nanometers of the reaction center. So coming through this completely, this 1 and 2 both will involved in this photophosphorylation. So it is gives the energy during this oxidation of water. The electrons move energetically down from water to an unknown substance Z scheme. So it relays the electrons from the Mn protein water splitting complex to the reaction center. So this oxidation of water, it requires the absorption of 2 protons by PS2 and 2 consecutive excitation of the P680 will lead to loss 1 of the electron at a time. So this is completely almost the electrons are passed through the 1 system to 2 system. The energy, so that is the assimilation power of energy will come this the ATPs and NADPH. So in this photophosphorylation, it occurs in the chloroplast because the light energy drives the protons and released into the thylakoid channel from the oxidation of water component of the PS2 and plastoquinone pool. So when cyclic electron flow from P700 to cytochrome B6, cytochrome F, that is the completely the plastoquinone is oxidized. So for this process, the one time the cyclic and non-cyclic will completion of this cycle. How many ATPs and NADPHs are formed? The assimilation power of the photophosphorylation is 3 ATPs and 2 NADPHs are produced through this photophosphorylation. So this is the very important. That means the it is also known as one chemiosmotic phenomenon, or it is also photophosphorylation involving the chemiosmosis. So that is also known as coupling factor. ATPase are coupling factors. So it is also in the one of the component, one of the complex of the electron transport system, ETS. So just to, while the assimilation power of the photophosphorylation completely, one cycle is completed in this way, 3 ATPs, 2 NADPHs are produced through the photophosphorylation in the light phase. So as I told you that completely the photosynthesis is completed in 2 stages. First one is the light phase. So light phase according to the photolysis of water and photophosphorylation, these all are undergo the light phase. So when light phase is completed, 3 ATPs, 2 NADPHs is the assimilation complete power of the assimilation power of the light phase. So this energy is utilized in the dark phase, Nana. How many ATPs? 3 ATPs, 2 NADPHs are formed through the light phase. So completely this assimilation power is utilized in the dark phase. They can produce some organic food, or they can produce the sugars or carbohydrates are formed in the dark phase. So completely the photosynthesis in the light phase, it is depending in the interdependent of the light and dark phase will completed, the photosynthesis is complete. So that is main cause of the first phase. This is the light phase. So in this electron transport system also, one of the major in the light phase. So in the electron transport system, completely the electrons move through the different 5 complexes are there. So in the 5 complexes, it is changes in the Q cycle. So it is also called quinone cycle. Quinone cycle takes place as completely. This is just, I will show diagram only, not go to depthly. So this is the chloroplast stroma in this PS1 and PS2. So pyridoxine, NADP reductase. This, the complete orange color is the chemiosmotic or ATP synthase. So in this, the protons H plus and H pluses are accumulated in this way. So this is the complete tail part. This is the head part. This is the ATP synthase. It will allow the protons to form the ADP plus inorganic phosphate to convert as a ATP. Okay. So that means the evolution of oxygen and protons during the photosynthesis. So the oxygen is generated in a 5-stage water splitting reactions. So it is mediated by some manganese containing protein complexes. So 5 different states, they are completely, they are driven redox reaction. Each transition requires 1 quantum of light, and each time 1 electron is transported. So in this, when excitation of chloroplast, it takes place with the short flashes of the light. The manganese ions are bound to the protein forming oxygen. So that is oxygen evolving complex. It binds the 2 water molecules to facilitate the oxygen formation. So this is completely the in the electron transport system. That means the requirement of the photons. Protons are, they are the no agreement with regard to requirement of photons to cause the photosynthesis. They can fix 1 molecule of the carbon dioxide. So this is the final statement, Nana. So completely the requirement of photons to cause the photosynthesis, and they can fix 1 molecule of the carbon dioxide. 3 ATPs and 2 NADPHs are needed for one CO2 fixed in the C3 plants. So while just in the next class, we'll discuss that the dark phase C3, C4, CAM cycles are there. So in the C3 plants, for one CO2 molecule fixation, how many ATPs and how many NADPHs are required? 3 ATPs and 2 NADPHs are needed per one CO2 fixed in the C3 plants. So completely 4 to 6 ATPs and 2 NADPHs are required per CO2 fixed in the C4 plants. So C3, C4, CAM, these 3 steps are involved in the dark phase. Okay. So for this is the assimilation power. 3 ATPs are needed to reduce 1 carbon dioxide to a simple carbohydrate. Okay. So, but slightly more ATPs are needed to form the complex molecules such as polysaccharides. So, need not to say here, just 3 ATPs are needed to reduce 1 CO2 to a simple carbohydrate. Okay. So, this is completely the electrons. The electrons are, they take the 2 electrons from PO5 and 2 proton atoms from the stroma and form the plastoquinone oxidized state PQH2. So which later is broken into PQ. The electrons are released to cytochrome B6F. That means, so which places ATPs are produced? That is the 2 completely PQ to PQH2 and cytochrome B to cytochrome F. So that is the formation of the ATPs. Almost you know that redox in NADP reductase. So that is the complete phase of the light phase. So non-cyclic photophosphorylation. You can transport the chain ETS, electron transport system. So excited electron, 2 electrons come from the PO5, plastoquinone, plastocyanine and cytochrome B6, cytochrome F. So the ATPs, completely ATP is produced in these complexes of that, and finally NADP also convert as a NADPH. So that is the non-cyclic. So yesterday Sal will just DCMU. So it is in between the P photosystem to plastoquinone. So DCMU also, it is the dichlorophenyl dimethyl ureanana. So it is an herbicide. It is also known as algicide. So it inhibits the photosynthesis and it blocks the plastoquinone binding site. Just you can observe here PQ, plastoquinone binding site. So of the photosynthesis too, it will disallowing the electron flow. So completely black this chain. Almost electrons are only they are situated in this PO5 area. So that's why it is allowing the electron flow from where it is generated. So in photosystem to plastoquinone, PO5 into plastoquinone to plastocyanine. So this is the DCMU also. It is one of the major factor to inhibit the photosynthesis, or it is interrupt the electron or proton transportation of the electron or electron transport chain. So this is completely the photophosphorylation and photolysis of water, and completely the cyclic and non-cyclic photophosphorylation. So up to here the light phase is completed, and next we will entering into the dark phase, Nana. In the dark phase, just I will share the screen now. Just wait.
Madam? Madam, please unmute yourself.
Okay, sir.
Okay.
Just okay, sir. Okay. So the next topic, that is the dark phase. So dark phase, diversity in carbon reduction pathways. So in that C3, C4 and CAM cycles. Today we are going to discuss about the C3 cycle. So the contents.
Please click on slideshow.
ఒక్క నిమిషం సార్. So sorry for the interruption. Okay, the C3 cycle. C3 cycle. So that is completely in the dark phase. Dark phase is involved in the 3 steps: C3, C4, CAM cycle. So in that we will discuss about the C3 cycle. The C3 cycle means plants in which CO2 is initially fixed as 3-phosphoglyceric acid. So that is known as the C3 plants. Okay. So in the C3 plants, the Kelvin cycle. So it is also known as a other names for the C3 plants or C3 cycle. So Kelvin cycle, it is also known as the dark reactions. It is also oftenly called reductive pentose phosphate cycle. So photosynthetic carbon reduction cycle. These all are the other names for the Kelvin cycle. Okay, Nana, dark reactions, Kelvin cycle, C3 cycle, reductive pentose phosphate cycle, and photosynthetic carbon reduction cycle. Simply PCR. So these are all names for only that is the C3 cycle. So in the C3 cycle, completely it occurs in the stroma. C3 cycle occurs in the stroma of the chloroplast. So it was observed that when carbon, that means carbon dioxide 14, generally kuda the 14 carbon dioxide was supplied in the dark reaction, the radio carbon dioxide. So that was supplied in the dark reaction. Phosphoglyceric acid increased. So, but there was a decrease in the concentration of the ribulose bisphosphate. So it's very important, RUBP. So when PGA increased and at the same time decreasing in the concentration of the RUBP. So this shows that the RUBP is the CO2 acceptor, primary acceptor of the C3 plants. That is the ribulose bisphosphate is the carbon dioxide CO2 acceptor during the formation of the phosphoglyceric acid. So completely the Kelvin cycle consists of the 3 main parts, Nana. So in that first one is the fixation, fixation, and second one is the reduction, and third one is the regeneration of the RUBP. Okay, the Kelvin cycle consists of 3 main parts: carboxylation or the fixation of the carbon dioxide, and second one is the reduction, and third one is the RUBP regeneration. Okay, so in this generally the stages of the C3 cycle. So this energy carrying molecules are made in the stroma of the chloroplast. So where the Kelvin takes place. As I told you that completely the Kelvin cycle occurs in the stroma of the chloroplast. So entrance point of view, stroma of the chloroplast. So the Kelvin cycle is not totally independent of light. So it releases on ATP and NADPH. So which are products of the light dependent reactions. So just now we'll discuss that one CO2 fixation. How many ATPs? 3 ATPs and 2 NADPHs are need for the carbon dioxide fixation. So that means it is completely the product of the light dependent reactions. So the light reactions of the Kelvin cycle can be organized in the 3 basic stages: fixation, reduction, and regeneration. So first we'll discuss that the fixation completely. So in this fixation, a molecule of ribulose biphosphate almost in the stroma, in addition to carbon dioxide, 2 other components present to initiate the light independent reactions, and they are the ribulose bisphosphate carboxylase, Rubisco. So it is very familiar in the C3 cycle. So the primary acceptor of the carbon dioxide, so that is the ribulose bis biphosphate carboxylase. So it is completely the CO2 is a high level of CO2 is, that is the carboxylase, or high level of oxygen is available, it is undergo the oxidization. So that means carboxylase and oxygenase both will presenting in the C3. That means it is presenting in the mesophyll tissue of the C3 plants or C3 leaves. Almost all the C3 plants, they have in the mesophyll tissue this enzyme, ribulose biphosphate carboxylase and oxygenase. It is very important in the C3 plants. So a molecule of ribulose biphosphate, it is carboxylated by carbon dioxide of the atmosphere. So an unstable 6-carbon compound is formed. So it splits into 2 molecules of the 3-PGA, 3-phosphoglyceric acid. So that is the main reaction. So RUBP have 5 carbon atoms almost. So it is flanked by 2 phosphates. So for each carbon dioxide CO2 molecule that reacts with 1 RUBP molecules of the 3-phosphoglyceric acid, 3-PGA from 3 carbon and 1 phosphate. This is the reaction, Nana, completely. So each turn of the cycle involves only 1 RUBP and only 1 carbon dioxide forms 2 molecules of the 3-PGA, 3 unstable intermediate compound. So otherwise the RUBP is the 5-carbonous compound. So it allows the atmospheric 1 carbon, both will combine to form the unstable, or that is completely unstable 6-carbon compound. So it is splits into 2 molecules of the 3-phosphoglyceric acid. So this reaction is regulated by ribulose bisphosphate carboxylase oxygenase enzyme. So that is the Rubisco. Rubisco enzyme is completely regulated this reaction or this first stage of the fixation. Catch it, Nana? Completely 3 unstable intermediate, that means a 6-carbonous compound. So in the presence of which enzyme? Rubisco, ribulose bisphosphate carboxylase and oxygenase. So this enzyme is the most abundant protein in the world, as I told you. So it is the key enzyme in the biomass production from the CO2 in plants. Okay. So it is catalyze the insertion of CO2 and simultaneous cleavage of the ribulose biphosphate into 2 molecules of the 3-PGA. Okay. And the second one is the reduction reaction. The second stage of the C3 cycle, reduction. So in this, almost the 6 molecules of 3-PGA. So the end product of the fixation. Sir, any? Three molecules are the 3-phosphoglyceric acid is reduced to 3-phosphoglyceraldehyde by ATP and NADPH. So they are formed in the light reaction. So whenever the power of the light reaction, ATPs are utilized in the reduction reaction second. In the C3 cycle, second step of the C3 cycle, reduction. Okay, Nana? 3-phosphoglyceric acid, it is reduced to 3-phosphoglyceraldehyde. Glyceric acid to glyceraldehyde. So in simple way, PGALD by ATP and NADPH, they are formed in the light reaction. So that is the 6 molecules of 1,3-diphosphoglyceric acid is reduced, and the molecules of NADPH to form the 6 molecules of the 3-phosphoglyceraldehyde. Okay, so this is the reaction. You know that. So 1,3-diphosphoglyceric acid plus NADPH2 and completely in the presence of triose phosphate dehydrogenase. So in this ATPs and NADPH both will utilized in this reduction reaction. So PGA is phosphorylated by ATP formed in the photophosphorylation. So it results in the formation of 1,3-bisphosphoglyceric acid. So it is catalyzed by the phosphoglycerokinase. And 1,3-bisphosphoglyceric acid is reduced to 3-phosphoglyceraldehyde by NADPH. So this reaction is catalyzed by triose phosphate dehydrogenase enzyme. Okay, so in the reduction, 2 steps are involved. In the first step, ATP